started. Um, Welcome. Thank you all for joining us today for the HMSC Research Seminar. My name is Yi-Chung Chung, and I'm the Academic Program Manager at Oregon State University's Hatfield Marine Science Center in Newport, Oregon. And I'll be your co-host of today's talk. But before we go into that, I want to go into logistics. And so we have your mics, cameras, and screen share off, and we ask that you keep them off for this event. Please ask any questions you might have using the chat box found at the bottom of or the top of your Zoom screen at any time, and we'll answer any questions at the end of this talk. We'll be recording this event, and it will be posted in a few days on the HMSC websites under the past seminar page, as well. It also ends up on our YouTube page. Just some quick announcements. Next week's seminar speaker on March 18th will be Samreen Siddiqui, a postdoc scholar from the Brander Lab in the Coastal Oregon Marine Experiment Station at Oregon State University. We'll be talking about contaminants of emerging concern in surface water with possible climate change impacts. If you need more info or the links to our upcoming events like Marine Science Day, check out our homepage and scroll to the bottom for more details. So with that, I'd like to introduce today's speaker, or at least let everyone know today's speaker has been invited by Jessica Miller, who's a professor in the Department of Fisheries and Wildlife and also a researcher in the Coastal Oregon Marine Experiment Station. And so I'm going to hand it off to Jessica for introductions.
Thanks, Yi-Chung. Good afternoon, everyone. And thanks for foregoing an hour in the sun to stay in and listen to this presentation. Zoe is a new postdoctoral scholar here at OSU, and she literally arrived, well, she started 10 days ago, so she's hot off the presses, so to speak, of having finished her dissertation and moved across the country during a pandemic. So Zoe actually started with her undergraduate here on the West Coast at Scripps College, where she actually did a senior thesis on the Channel Islands, and then moved off to the Midwest and worked on those Great Lakes, the inland seas of the US, getting a master's of science at Purdue University, and then moving on to her doctoral research at Ohio State. And she has focused, her master's research looked at the effects of hypoxia on fishes in Lake Erie. And her dissertation, which she's going to tell us about today, looked at effects of early life conditions on Lake Erie Walleye. And she'll be working with us, Ben Laurel and myself, on similar questions on Pacific cod in the Gulf of Alaska. So we're happy to have her, and I will hand the controls over to Zoe. And thanks again, Zoe, for presenting.
Thanks for the introduction, Jessica. And thanks everyone for being inside and watching this, unless you're watching it outside, and then hopefully you're enjoying both. But yeah, this is basically kind of what I presented as my defense seminar for my dissertation. But yeah, thank you for your interest in my research. So the traits that an individual exhibits, the things like their size and morphology and behavior are due to a combination of legacies. So individuals have first their genetic legacies, so the genes that are passed down to them from their family tree. They can also exhibit epigenetic or parental legacies if the experiences that their parents have affect the way that their traits are expressed in their life. And then individuals also have experiential legacies, where the experiences or environments that they've encountered earlier in their life continue to affect the traits that they exhibit throughout their life. And my research primarily focused on these experiential types of legacies. In particular, my research focused on experiential legacies from early life experiences. And we focused on early life because early life can be quite influential to an individual. Broadly, I define early life as any time prior to maturity, but individuals in early life might be more vulnerable. They could be more resource limited than adult counterparts, and they are still developing important structures that adults have. And because of all of this, individuals might be more likely to not survive through this early life period. However, for individuals that do survive, to adulthood, the traits that they express might still be influenced by those experiences that they had in early life. And just a little vocab, when I talk about experiential legacies from early life environments, I'm including things like latent effects and carryover effects in that definition. And it's important to understand these individual traits because the traits of an individual scale up to population consequences. So when individuals experience different early life environments throughout a population and are able to survive those conditions, those experiences might continue to inform the traits that the individuals express within the population, thus affecting a variation within a population and potentially some population-wide traits. Additionally, individuals that share similar experiences, like individuals that were all born in the same year, for example, might share things like, might continue to share those traits throughout their life, creating something like cohort effects that you can see at a population scale. And so there's value in examining these experiential legacies from an individual to a cohort to a population scale. And that was the overall objective kind of my research, was to look at how experiential legacies affect individuals and then scale that up to a population level. And I did that with a few different aims. The first one was to look at experiential legacies across species and populations using a meta-analysis technique. And then I also looked at experiential legacies within a focal population, Lake Erie Walleye. And within Lake Erie Walleye, I looked at experiential legacies at the individual level with laboratory experiments that manipulated early life diet quality, at the cohort level using long-term data analysis, and at the population level using demographic modeling. Now, Lake Erie Walleye allow us to ask some really interesting questions related to experiential legacies for a couple different reasons. One of which is that Lake Erie Walleye are pretty sensitive to their early life environments. And so this figure here shows the Lake Erie Walleye recruitment for the past couple decades for each annual cohort. And we associate the years of really high recruitment events with good early life environments and the years with poor recruitment with poor early life environments. And you'll notice that more recently we've had actually like a couple really good years of recruitment, which is great for Lake Erie Walleye. They can live up to 20 years in the lake, and so even 1 year of really high recruitment can sustain the population for decades. However, prior to this period, we had an extended time when we had relatively low recruitment in the lake. We often assume that cohorts from these years where we have relatively low recruitment provide little support to the population overall, given their numerical abundance in the population. However, I was interested with if we can consider experiential legacies for these cohorts, if we might better understand the role they play in the population. So if, for example, you could have a situation where their early life experiences actually allow them to buffer the population more, if those early life experiences allow them to respond better to poor conditions later in life, which could buffer the population when high recruitment events are rare. Alternatively, they might be even less beneficial to the population than we think based off of their numbers if they're continuing to be negatively affected by poor early life experiences. And if it's the latter case, then these few years of really high recruitment might be even more important to the population, or understanding the population overall. Another reason we were interested in looking at experiential legacies with Lake Erie Walleye is because there's been some changes to their early life environments, and we're not exactly sure what those consequences might be. So this is a larval Lake Erie Walleye, which is an obligate zooplanktivore, meaning that changes to zooplankton or phytoplankton could dramatically affect the food quality that these very sensitive life stages experience. And with climate change, we're seeing changes in temperature and eutrophication, which can directly affect all of these different trophic levels. But we might also see a situation where changes to phytoplankton are scaling up and affecting the food quality that larval walleye are experiencing. However, before I get into how we looked at experiential legacies with walleye, I want to talk a little bit about the general patterns that we tried to identify in experiential legacies across species. So one of the reasons I wanted to look at some of these general patterns is because there's a big variety in experiential legacies that exist. So for example, you can have a situation where an individual exhibits somewhat of a trajectory response. So say you've got an early life environment that is objectively poor or good. So say you've just got low food abundance and high food abundance, and that's going to affect an individual's early life phenotype. However, you can get a situation where regardless of the later environments individuals encounter, their later life phenotype continue to reflect some of that early life environment that they experienced. And so you can get this lifetime trajectory-like response. However, you can also get a situation where individuals are able to compensate for their early life experience. So you've got, again, a poor or good early life environment. However, something about those later life experiences allow individuals to compensate. And so their later life phenotype is indistinguishable regardless of their early life environment that they encountered. And then you can have a situation where individuals demonstrate something like phenotype environment matching, where the later phenotype performs best when the early life environment matches that later life environment that they experience. However, if those early life and later life environments do not match, then the later life phenotype will perform not as well. And so given this variety in the different types of experiential legacies that exist, it can be hard to understand what phenotypic response you might see, how the magnitude of that response, and whether or not that phenotypic response will benefit or detriment an individual. And one of the reasons for this might be that experiential legacies can affect fitness in a couple of non-exclusive ways. The first one is that the experience, when an individual in early life experiences some challenging conditions, they might reallocate energy away from functions like growth and development towards survival to allow them to get through that period of life. And in doing so, that might put them on a trajectory-like response if they have to continue to sort of pay the debt of that reallocation of energy from early life experiences. However, individuals might also be able to modify their phenotypes based off of those early experiences to allow them to increase performance in future conditions, kind of like reprogramming them. And that might be how you get something like phenotype environment matching. So I've been talking a lot about experiential legacies across things that are related to body conditions, so like size and growth. However, these experiential legacies really occur across all types of phenotypic responses. You can have experiential legacies related to learning, longevity, movement, and reproduction. And additionally, the direction of the effects of these might not all be in the same direction. So you can have a negative consequence on body condition, but a positive response, positive phenotypic response related to reproduction. So with this first aim, my general goal was to try to identify some patterns in direction and magnitude of experiential legacies and see if we could kind of explain the variation that we see in those. And the approach for this was a meta-analysis, where I specifically looked for studies that caused early life stress due to nutritional restriction. And in particular, I looked for studies that had treatment control pairs. So treatments where early life nutrition was poor or good that affected early life phenotype. And then the later nutrition was standardized to some general good level to allow for recovery if that was possible. And then the later phenotype was measured. And so overall, I found 81 studies that we could include based off of our criteria. That was across 65 different animal species and included 683 of these phenotypic responses. And these responses occurred across all 5 of those different categories that I had mentioned earlier. Now, I'm not going to go into specific results just for the sake of time today, but in general, we found that these trajectory responses, where the early life environment ended up continuing to affect individuals' later life phenotype, these types of responses were quite common in what we saw with our results. And this result is consistent with that first mechanism I talked about, where altered resource allocation in early life continues to affect individuals as if they're sort of paying some energetic debt throughout life. All right. Now I'll talk a little bit more about what we saw with experiential legacies with walleye. So again, I looked at experiential legacies at an individual scale, a cohort scale, and a population scale. But I'll first talk about our how we looked at experiential legacies in individuals. So as I mentioned before, we're not exactly sure what the consequences are of some of these changes in the early life environment in Lake Erie are. And we're particularly concerned about how temperature eutrophication might be affecting phytoplankton because the temperature eutrophication can affect the way that phytoplankton produce important nutrients like fatty acids and vitamins that could scale up to affect the food quality that walleye are experiencing in early life. And so we were particularly interested in how temperature eutrophication might be affecting the production of polyunsaturated fatty acids, or PUFA, as I'm going to call them throughout the talk, and vitamin E. And so we focused on PUFA because these are essential fatty acids, and they often can't be synthesized by fish. They actually have to be consumed from their food. And we focused specifically on EPA and DHA. And that's because these are important to early life stages and during development. And we were also interested in a form of vitamin E, alpha tocopherol, which is seen to be important for absorbing high PUFA concentrations in diets. So our experiment consisted of two phases. The first phase, which was diet modification, and this started immediately when larvae started feeding. We modified the different treatment diets for individuals to see in a couple different treatments. Our first treatment was a control, which was low PUFA control. And then we had a high PUFA treatment, which was high in EPA and DHA. And then we had a high PUFA and vitamin E treatment. And we had 3 replicates for each of these different treatments. And so after this 10 days of feeding on these diets, we switched all of our individuals in our treatments to a standardized diet in which a high quality food was provided equally to all of the treatments for 27 days. And we expected that we would see something like this trajectory response, given how common it was in our meta-analysis results. And so here we thought that our early life environment, that was going to be our phase 1 early life diet, where our control is our poor food conditions and our 2 high PUFA treatments are our good early life environments. And we thought that we would see that reflected in the early life phenotype by being integrated into the body tissues and affecting the performance of individuals. And by performance, I'm mostly talking about size and growth. And then after that 10-day feeding period, individuals were exposed to that standardized diet. However, we thought that their later phenotype would still be affected. So we thought that that early life diet would continue to be reflected in the tissues, as well as continuing to affect the performance of individuals. All right, so our results from phase 1, after feeding on 10 days of the different dietary treatments, we found that those diets were incorporated into the tissues. We found that the fatty acids and vitamin E within the fish tissues were reflective of the diets that individuals have been fed for the past 10 days. And we did see an effect on performance as well. This figure here shows density on the x-axis and mass on the y-axis. And right now, these the points right here are tank average mass means. For just the white dots are for our control treatment, with the error bars indicating variation around the tank means in mass. And so our white dots here are the control, the teal dots are the high PUFA treatments, and the orange dots are the high PUFA and vitamin E treatments. And we found a significant effect of diet as well as density on mass in the tanks, with density having a significant negative effect, and diet being significantly, diet affecting mass by being significantly larger individuals were found in the high PUFA treatments compared to the control. So far, we're meeting our expectations. We did find that the diet was integrated into the tissues and affected performance. So now let's look at our results from phase 2. So at the end of phase 2, we actually ended up finding that the tissues were not, that the biochemical composition of the tissues were not reflective of that phase 1 diet, and instead they reflected the phase 2 diet. So individuals were able to absorb the fatty acids and vitamin E that they were receiving during that standardized diet, which was reflected in their tissues. However, we did continue to see a lingering effect of that early life diet on body mass, on performance. So again, we have density on the x-axis and mass on the y-axis. And we found that that phase 1 diet was still significantly affecting mass between our treatments, and density still significantly negatively affected mass in our treatments. And again, the diets were, our 2 PUFA treatments were significantly larger than our control treatment. So overall, we did see evidence for this trajectory response. We did see that those early life dietary conditions affected the performance of individuals' phenotypes later in life. We did not see that that early life diet continued to affect the tissues, however. And so because of that, that could be that individuals might be compensating, be able to compensate for that early life experience. And so if we had looked at growth metrics or size metrics for a longer period of time, we might see that those individuals were able to compensate and equalize their sizes a little bit more. However, even if they were able to compensate over the long term, we did still find that there was a lingering difference in performance for that period of time. And for larval fish, that might be really significant for their survival. Since smaller fish tend to be more susceptible to predation and other sources of mortality, they may not survive to actually be able to compensate. All right. So next I'll talk a little bit about what we found when we looked at experiential legacies at the cohort level with Lake Erie Walleye. So as I mentioned before, when individuals have common early life experiences, like all being born in the same year, that might create a cohort effect that is reflected in the population throughout life or throughout the population. And I chose to look at cohort effects with size and growth data on walleye, since size and growth is really important for walleye survival, reproduction, and harvest. Additionally, walleye are indeterminate growers, meaning that they don't stop growing once they reach a specific size or age. And so we weren't exactly sure as how those early life experiences would affect things like size and growth in an organism that doesn't stop growing at a certain size or age. But there's a lot of different things that affect growth from one age to the next in an individual. So you can have things like the recent environment, things like temperature and prey availability affect growth. But the growth that an individual experienced in the previous age can also affect growth in the current time. And there can also be an indirect effect of the size that individuals achieved in early life or the densities that they experienced. And since size and density in early life can be affected by those early life environments or maternal environments, there could also be an indirect effect of those environmental conditions on growth. And then those early life and maternal environments can also have a direct effect on growth if those early life environments, if either of those environmental conditions affect selection or create a provisioning or parental legacy that might look like a direct effect on growth. And in particular, I was interested in how these 5 different pathways potentially affected walleye cohorts, growth in walleye cohorts. I was interested in that direct effect of the recent environments, the effect of growth in the previous year on growth in the current year, and then separate effects of density in early life and size in early life, as well as the direct effect of maternal environments. And so for the growth data, we calculated growth as an annual cohort change in length, which is the difference in a median length at age, but we also corrected that for size. So it was a growth length residual, really. But we got the data for this growth metric from annual gillnet surveys conducted by the Ohio Division of Wildlife for the past few decades. And in particular, we focused on growth during ages 3 through 5. And part of the reason we did that is because growth at that time period is still relatively rapid, so it might still be susceptible to changes in their environmental conditions. but also growth during ages 3 to 5 is relatively linear across cohorts. So for the data that we got for the potential predictor variables, for the recent environment, we looked at the effect of prey, temperature, and harvest pressure on growth. And that's because adult female walleye, their condition tends to be, has been shown to be responsive to their temperature and competitive pressures. So we thought that those factors might also affect growth in adults. And then we also looked at the direct effect of growth in the previous age on growth in the current age. And that's because walleye are capital breeders, so they're contributing energy to reproduction and growth continually throughout the year. And so the way that individuals contribute energy towards growth or reproduction in the previous year might affect the way they do that in the next year. And we calculated this growth in the previous year the same way we calculated growth in the current year. And then we used the cohort density in August when individuals were in the first August of life. And that's because differences in density between cohorts are relatively established by that period, since a lot of the environmental changes that cause big mortality events have occurred. And so mortality is relatively stable after this period. And we looked at size in early life from that same period, looking at median length at that August period of time. For similar reasons, because a lot of the environmental conditions that would have either caused selection events or affected whether individuals were fast growers or slow growers would have already occurred. And then for the direct effect of the maternal environment, we looked at the effect of percent ice cover that mothers experienced right before hatching during the year that cohort started. So walleye hatch out in April to May, but we looked at ice cover in February to March of that same year because data has shown that ice cover experienced by mothers right before spawning affects maternal body condition as well as egg quality in walleye. And we thought that might continue to have an effect, a provisioning legacy or some sort of a direct effect on growth with walleye after that. So our approach for this was to use confirmatory path analysis. Specifically, we used piecewise structural equation modeling in R. And we created a series of candidate models that had different combinations of those different pathways that I was talking about. And then we evaluated those different candidate models using model selection with AICC. This table here shows the different models that we created with the different pathways being represented at top. And you'll notice that we didn't include every possible pathway combination with our models. We only included pathways that we thought would made sense given what we knew about walleye biology. And you'll also notice that we had a null model that didn't include any of the pathways. However, this null model did include factors related to the biological condition of the cohort, so things related to sex and proportional maturity. So this model here is the first model, which includes all possible pathways. And I'm just going to kind of talk through what these different arrows mean. So our first pathway was represented by a direct effect of temperature, prey, and harvest on growth. And then our second pathway we represented with a direct effect of growth in the previous year and growth in the current year, as well as an effect of growth in the last year on proportional maturity in the cohort in this year. And then we also looked at an indirect effect of age 0 walleye density by looking at that direct effect on growth in the last year. And we did the same thing with age 0 walleye length. And then our fifth pathway was represented by a direct effect of ice during the year of hatch on growth. And then the rest of the relationships in this model were factors that were included in all models that were related to the biological condition of walleye. So our results showed that our most supported model was our 12th model, which included our the second pathway, which was growth in the previous year affecting growth in the current year, and our fifth pathway, which was the effect of maternal environments, specifically percent ice. And I'll walk through what model 12 kind of looked like here piece by piece. So the growth in the last year had a significant negative effect on growth in the current year, meaning that years where growth was higher than average were followed by years where growth was lower than average, and vice versa. We also found a significant positive effect of ice from the year of hatch on growth in the current year. So that means that years where the cohort was hatched out when there was a greater amount of ice cover on Lake Erie, 3 to 5 years later, that cohort had faster growth than a cohort that had less ice during the year of hatch. Now, the rest of the relationships in this model and the strongest relationships were those related to the biological conditions, which kind of makes sense. We knew that those would be some of the strongest factors affecting growth. So overall, at the cohort level, we did find that there was a cohort effect, but it was due to an epigenetic or provisioning legacy based on that experience that mothers had right before spawning. So we're seeing that ice cover affects, we knew ice cover affected maternal condition and egg quality, but we're seeing that effect can persist 3 to 5 years later and be reflected in cohort-based growth. This might be a bit concerning with climate change, since we are seeing warmer winters. And in particular, this January, we set a record, the Great Lakes had the lowest percent ice cover that it's had since they've been reporting this information. We don't know how long this cohort effect persists. We saw it 5 years later, but there's other factors that could mediate this effect and cause it to be less significant. And one of those factors might be the other significant pathway in the model, which was that trade-off we saw between growth and adjacent years. So I'm going to talk a little bit about the results we saw here with this figure here, with age on the x-axis and growth on the y-axis. And that gray line across there is the average cohort growth rate, across cohorts for ages 2 through 5. Now, if we'd seen a trajectory response, like we'd seen in some of the previous bits of research that I've presented, we would expect that cohorts that had faster than average growth at age 2 would continue to have faster than average growth, and cohorts that had slower than average growth at age 2 would also continue to have slower than average growth. However, if we saw a indication of some compensation, what we'd see is that cohorts that had faster than average growth one year would have slower than average the next, with a lot of sort of crisscrossing of this average line. And when we look at actual data, these data here are for males, with all of the black points and lines indicative of cohorts that had faster than average growth at age 2, and the gray dashed lines are for cohorts that had slower than average growth at age 2. And what we see is this compensation response, where we see continual crossing of that average line, which might indicate that there's some compensatory growth happening. However, our data were on cohorts, and so it would be hard to actually say compensatory growth is happening. We really need to see data on individuals to definitively say that. So overall, we previously saw some evidence for trajectory responses in individuals. We're seeing some evidence for compensation at the cohort level with Lake Erie Walleye. So now let's talk about how we looked at experiential legacies at the population level. Given the diversity of experiential legacies that exist, that might mean that different experiential legacies are beneficial at a population level under different circumstances. So for example, different combinations of early and later life environments might make some experiential legacies more or less beneficial in those circumstances versus when early life and later life environments differ in some different way. So whether early life and later life environments match or they differ in different directions. Additionally, changes in certain life history characteristics, like things related to longevity and reproduction, might make experiential legacies more or less, different experiential legacies more or less beneficial to the population. So the goal of this aspect of my research was to examine the scenarios under which these specific experiential legacies are most advantageous at the population level. And so I did this by looking at combinations of good and poor early life environments, as well as by looking at this across three populations that differ in key life history characteristics. And I looked at these different scenarios with 4 different experiential legacies. So the first one I looked at was with a control where there was no experiential legacy. So individuals within the population only respond to their most recent environments. So early life phenotypes are reflective of that early life environment, and later life environments affect that later life phenotype, but there's no relationship really with the early life environment and the later life phenotype. Next, I looked at the experiential legacy of lifetime trajectories, where those early life environments establish some lifelong responses. And so we've been seeing this, I've been kind of talking about this throughout this talk, but this is a scenario where early life environments establish that early life phenotype. And then regardless of the later life environment that the population experiences, that later life phenotype is going to be more reflective of that poor early life environment experienced. And I anticipated that this type of experiential legacy would be most advantageous at a population level when that early life environment was good and the later life environment was poor, since the population could be able to buffer those poor later life environments if they'd had that good, if they have a good chance of experiencing a good early life environment. The next experiential legacy I looked at was environmental specialization, where individuals perform the best in the same environment that they experienced in early life. And so for this one, if early life environments and later life environments are mismatched, their later life phenotype will perform worse than if their early life environments and later life environments are matched. And so I thought this would be most advantageous to populations when that it's likely that those early life environments and later life environments will be matched. And the last experiential legacy I looked at was later stress resilience, where the poor conditions in early life allow individuals to respond better to later life, to poor conditions later in life. So in this situation, if the population has, different populations experience poor or good early life environments, and then universally there's a poor later life environment experienced, the population that experienced a poor early life environment is going to be better prepared for that. And so their later life phenotype will reflect this. And I thought this would be most advantageous in this situation where early life environments were likely to be poor and later life environments were likely to be poor. However, I should note that there was no penalty like with the mismatch environments. So that if early life environments were poor, there was no penalty if later life environments were good. They were able to compensate for that early life experience. So my approach to this was using matrix modeling, where we assumed that all individuals responded similarly to a sequence of high and low environments. So that means that the responses are cohort based. So that just means that all individuals respond the same way. There's no inter-individual variation. So if poor early life environment is poor, all individuals respond the same way, and same as if the early life environment is good. And this here is a schematic of the model description. And so we had 5 different stages with 2 different juvenile stage possibilities. The only reproductive stages were medium adults and large adults. And so I'll walk through kind of what all these arrows mean here. But the purple arrows here that are highlighted represent fecundity and early life survival. And so the early life environment affects how many individuals survive that in early life, but it also determines whether the population contributes individuals to the 1P group or the 1G group. And so if early life conditions are poor, the population contributes to the 1P group, whereas if early life conditions are good, they contribute to the 1G stage. So the early life environment affects both survival to the juvenile stage as well as whether individuals enter the 1P or 1G classes. These arrows here represent the probability of individuals in each stage growing to the surviving and growing to the next stage. And you'll see that our 1P and 1G juveniles grow to the subadult stage. However, they can also grow fast and grow straight to the medium adult stage, at which point they would be reproductive. And we determined whether or not the individuals would grow fast or slow to that juvenile stage by a combination of their later life environments that they experienced, as well as the experiential legacy that they were modeling for us. And then these arrows here indicate the probability of individuals surviving and staying in the same stage. And so all classes can do that except for the juveniles, which have to grow to the next stage, to either stage 2 or stage 3, if they survive. So then we looked at our environmental scenarios with probabilities of just generic good environments and with 5 different probabilities. And so for early life, we had the probability of a good early life environment or a poor early life environment. based off of these values. And then the same thing with the later life environment, but they didn't have to be the same. So we have these 5 different combinations of early life and later life combinations that we focused on for this right now. And the 4 of them were the most extreme values. So very unlikely to have a good early life environment and very likely to have a good early life environment mixed with every combination, mixed with very likely to have a good later life combination and very unlikely to have a good later life experience. And then I also had a intermediate environmental scenario that we thought was more reflective of what Lake Erie Walleye might be experiencing, since right now it's not very likely that they'll experience good early life environments, but it is quite likely that they will experience good later life environments. And so as I mentioned, we looked at this with our focal species, Lake Erie Walleye, and they represent a periodic life history, meaning that they have high fecundity and older age at maturity and generally low juvenile survival. However, we wanted to see how our walleye population experiential legacies compared to populations with different life history strategies. So we also modeled an equilibrium population and an opportunistic population that differed in their combinations of fecundity, age at maturity, and juvenile survival. And for analyses, we looked at every combination of experiential legacy and environment and life history, ran the model for 1,000 time steps, and then did that 100 times. And today I'll only talk about one of our responses, which was the population growth rate, or lambda. And so I'm going to be showing you our results on figures that look like this, with our environmental combinations on the x-axis, and on the y-axis is our geometric mean lambda, which is our population growth rate. And there's going to be this bar across at 1, because 1 means stable population growth rate, meaning the population isn't growing or shrinking. Values below 1 mean the population is shrinking, and values above 1 mean the population is growing. And right now I only have plotted on here our experiential legacies for that, the intermediate environment. However, when early life environments were very likely to be poor, population growth rates were very low. Whereas when the early life environments were likely to be good, were more likely to be good, the population growth rates were much higher. And so we're seeing that that early life environment is really driving our population growth rate. And we actually see that across our life histories. In both our equilibrium and opportunistic populations, we see a similar pattern. However, if we look in at our experiential legacies that we see within each environment, we see that we do have this lifetime trajectory response being significantly more beneficial to the population growth rate than the other experiential legacies. And we actually see that trend across different life histories as well. So at a population level, we see that those early life environments, first of all, seem to be extremely important to population dynamics across life histories, at least to population growth rates. And we again see this trajectory response being common across our different life histories, at least when there's specific environmental conditions that are met. So we see that this trajectory response can be quite beneficial to a population regardless of life histories, when that early life environment is likely to be good, but the later life environment is likely to be poor. All right, so sort of wrapping everything up. We're across the different analyses that we looked at, we see that this trajectory response seems to be relatively common, both across species as well as within Lake Erie Walleye. We do see this response, where that early life environment is not only affecting the most immediate phenotype, but seems to have some lingering effect on individuals' phenotypes. If this trajectory response really is dominant across walleye phenotypes, that might mean that these years that have really poor recruitment provide little support to the population, even less so than their numbers actually imply, as they might be continuing to experience negative effects from those early life environments that they encountered. And that would mean that the overall population, both quantity and quality, is really supported by these years with really high recruitment, which might be concerning if those years are becoming less frequent. However, we also did see some evidence for compensation in walleye with that consistent size and age that we were seeing with cohorts. And so if compensation is really affecting population, then what we might be seeing is that these cohorts that have really, that are relatively small in the population might actually be able to buffer the population a little bit better than we thought, despite their low numbers, if they're able to compensate for those poor early life conditions they experience. However, before we can actually say anything about that, we need to understand a lot more about individuals. We need to know if compensation, like compensatory growth, is actually happening at an individual scale. And we also need to know those trajectory responses that we saw given early life diet, how long those last as at an individual level, and if that affects mortality rates. And then we also need to know if there's some trade-offs or costs, because things like compensatory growth tend to be associated with trade-offs with longevity or reproduction. And then once we understand those factors at an individual level, we need to scale that up and understand them more at the cohort and population scale. So overall, with my research, I think experiential legacies are an important factor to consider. And it's even more so considering how fast environmental conditions are changing. If we need to understand the early life and later life environment to understand what later life phenotypes we might see or how that's affecting population dynamics, it's going to be a lot harder to do that if these environmental conditions are changing more rapidly than we can account for. So with that, I'll thank, first of all, everyone who was involved in this research. Since this was my dissertation research, I had a lot of help from various mentors and collaborators, as well as a lot of help from other grad students and technicians, as well as different funding sources. And with that, I will take any questions anyone has.
Great. Any questions from our audience? Thank you, Zoe. And I'm going to go ahead and post information for the recording of this seminar in case folks want to access it.
Thanks, Zoe. That was a good overview of a lot of work. I imagine you probably could have talked for the full seminar on both the first and the last part. So I do have a question. It went kind of quickly. And so the life history comparison you made with the opportunistic and the periodic and equilibrium species were really interesting. Yeah. And when I think of how life histories evolve, you know, one of the ideas is it's responding to the scale and strength of environmental covariation to the life history development. So did you take that, or did you think about that in the modeling framework, and how did you incorporate those different life histories?
So you're asking about like how the different life histories evolve and stuff? Or
yes, right. How you were reflecting them in the model. I missed that piece.
Yeah, so the model schematic that I showed you was just for walleye, really. I ended up creating slightly different models for the different life histories. So for example, with bay anchovy, they were actually able to reproduce at that first stage. So we had and we had their time step was different because since they become reproductive, they become mature so quickly. Our time step was actually 4 months instead of a year. And so I modified the model like in a couple of different ways to try to account for the fact that those life histories are so dramatically different. But we wanted to keep the general structure of the model with the 5 stages the same to make it a little bit easier to have those comparisons. So I tried to sort of like negotiate that, given that we based the model originally on walleye, but also modified things like which stages were mature and how much fecundity each of the different, like, because the fecundity was size-based or like class-based, and so that all differed depending on the different life histories that we included.
Thank you.
There's a question from Tom H. We think of good environments as being a suite of conditions being good. but bad environments can be bad for multiple reasons. How do you think that different bad environments might affect thinking or modeling this phenomenon?
Yeah, that's a terrific question. Basically, talking about generic good or bad environments is a huge simplification that I'm definitely aware of, because ultimately these are multivariate stressors. And so, for example, higher temperatures than an individual is used to is going to affect an individual different physiologically than hypoxia or than ocean acidification or different things like that. And so I think the way I was thinking of it was sort of just some generic framework to talk about just like good and bad experiences, thinking mostly about their effect on that early life phenotype more. So like, does it affect the phenotype, and does it cause like a physiological stress that affects that phenotype? But I think that it definitely could be expanded to sort of look at different ways that that phenotype could be affected, whether it affects growth or development, like it could affect those in completely different ways, or different physiological functions that we don't see until later in life too. So I think that this is definitely like a simplified version of thinking of this, but I would be excited to think about ways that you can actually modify some of these models or some of these ideas, to look at, okay, the environment is bad in this way, or we're like modifying this environmental condition which would have this effect on the phenotype. I don't know if I answered your question, but I rambled a lot. So
He says, great, thanks. Zoe, I had a question. So in looking at the early life stages of the walleye, this idea of ice cover having kind of a what appears to be a positive effect. Can you elaborate more on that? And I'm sorry if you did talk about it and I missed it, but I would love to hear more about that.
Yeah, I totally skimmed over that. But yeah, so ice cover. So my background is in perkids, so I know that this is definitely different for depending on the species that you're looking at and everything like that. But with perkids, so things like yellow perch and walleye, the thought is that ice cover is really important, because it allows mothers to decrease their sort of energetic need, their metabolic rates over winter. And we're not exactly sure, because you could also say like as long as they have enough food available, it's what might be hypothetically okay to have a warmer conditions as long as they're meeting that metabolic needs. But maybe since we're seeing a negative effect, that means that there's not enough food available. But basically, we think that there's a couple different ways that ice cover can affect those early life stages. One is by that maternal effect, by if there's conditions are cooler, they can slow down their metabolic rate, and so they can actually have a higher condition and provision their eggs better when they reproduce. So that's one way. And then the ways that then there's other ways that it affects environmental conditions, by affecting the phytoplankton community that's available, or by affecting other lake conditions, other lake-wide conditions. But the direct effect through mothers would be through affecting how much they can contribute to their eggs in the spring.
So that they're less active and contribute more energy to producing better eggs.
Yeah.
So kind of almost a hibernation effect.
Yeah, I mean, not quite, because definitely ice fishing in the Midwest is huge. And so you definitely, especially for perch, which also have this similar phenomenon. And so if you put food in front of them, they'll eat it. They don't hibernate that much. So if you put a big fat worm in their face, they're gonna eat it.
All right, thank you. Any other questions from online? Oh, here we go. Will has a question. If winter ice cover continues to diminish, how do you think cohort population growth will change?
Yeah, that's a great question. I mean, I know a lot of people who are doing like overwintering studies with female perch and female walleye, to kind of see what sort of effects that has on a physiological basis. with the idea that we can maybe scale that up to understand cohort and populations. I know that in parts of the Midwest where they're seeing consistent warmer winters, they are seeing declines in walleye populations, and in fact, like a shift in the community towards things like largemouth bass, which like warmer conditions. But it's kind of hard to tease out that ice cover effect because you're also simultaneously having land use change and differences in nutrients and what these different species like. It's sort of like a whole regime shift in some of these lakes that we're seeing that. That being said, though, Lake Erie walleye have never been like more abundant. Like we're having huge population abundances, and so I think it's something kind of like Tom kind of alluded to, that there's this combination of environmental conditions, and we might get to the point with ice cover where we push the population too far. But until then, Lake Erie walleye seem to be doing really well. That it's quite different though from other populations in the Midwest, and it's just a very different system. So there's a lot of different factors that are affecting it. So ice cover is one, but so is turbidity, like how much dirt is in there and how much nutrients and things. So it's a lot going on. I think eventually if we lose enough ice cover, we will lose walleye populations. Like I think it's that dire, but yeah.
All right, are there any other questions? Oh, one more, here we go. I might have missed this. Tom asks, in the lab experiment, did you intentionally vary densities, or was that just an effect of variable survival in the replicates?
Yeah, we did not intentionally vary the densities. What actually happened was, so given that larval fish are like little basically pieces of string that can very easily die, before we even started we had like transferred the larvae into our tank systems. And before we'd even started their dietary treatments, we had a big mortality event that was not equally distributed across the tanks. And so we decided to, at the end of the experiment, to consider density as a covariate. And based off the mortality that we saw, we distributed our dietary treatments so that they would be sort of like evenly crossed across densities. So we didn't have one treatment that was in our like only in our most dense tanks. And so it was kind of like an afterthought of like, we're going to need to account for this density effect. However, when I looked at like comparable experiments, density is not often reported, and there's sort of like density was between this and this, like sort of like, you know, we're okay. But given that we saw a big effect of density, I actually kind of wonder if some of the variation that other studies have seen could be accounted for if they like measured their tank densities actually. But
Tom says, been there, nice way to respond to the mortality event. All right, well, we are at 4:30, and I do want to be respectful of everyone's time. If you have any other questions, Zoe, would you mind putting your contact information in the chat for folks? And you're getting accolades online. Well, thank you again, everyone. Thanks again for joining us today. And Zoe, thanks again for speaking at our seminar today. And welcome. Welcome to Hatfield. Welcome to the campus.
Thanks, Yichun.